Hi, I am Laura and I will be talking about storing bioimage metadata with Amero. Amero is a client-server software for visualizing, managing and annotating scientific image data. Amero is free and open source. If you don't have an institutional server, you can visit the Amero web pages and sign up for a free demo. Here is an example of a project in Amero called Dictee. Let's take a closer look. So we have a project called Dictee containing a dataset. If you are familiar with the REMBI guidelines, we will be treating the project as a study and the dataset as a study component. If you are not familiar with REMBI, you may want to check out the other RDM bytes on that topic. In this example, I have data imported into Amero already. However, you can create a new project and dataset and add metadata to it before you have any images. You can then just add your images to the appropriate projects when you have acquired them. For the study metadata, I have added the study description to the project details and added the general dataset information to the project key value pairs. Next, we will look at the dataset containing the images. So I have the study component title. Note that I've started it with a date in ISO format. This is because I want to order my datasets in chronological order. I've added a description to the dataset details. You can see here that I have noted that the analysis results are attached to each image rather than to the dataset. This is so that if other users come to this dataset looking for the results and find they aren't attached to the dataset, they know where to look for them. I then use the key value pairs for all other metadata, starting with the imaging method. Next, I consider the biosample metadata. I've used a key with no value to add a heading. You may prefer not to do this and just have the metadata. Either way is fine. Just make sure to cover all the metadata modules described by REMBI. I have also included the biosample identity in the study component in this case, because all the images are of the same sample. You may choose to move this to the individual images or include it in the image file's name if the images come from different samples. The same goes for the specimen metadata. If you have control and test specimens in the same dataset, you may wish to add this metadata to the individual images instead. For the image acquisition, you can see I have included very few details. This is because Amero is able to read information from the microscope metadata when the images are imported. We can see this under the Acquisition tab for each image. Since I know these details are recorded automatically, I don't bother duplicating them elsewhere. Going back to the previous slide, you can see all I've added here is the instrument name and the filters used. If we look at one of the images, we can see that the image data metadata has been automatically read into Amero on import. So once again, I don't bother to record this information elsewhere. No image correlation was carried out for this data, so I skip this part of the REMBI metadata framework. But you can see here that this image has one region of interest, and this is part of the analysed data. Since all my images were analysed using the same method, I include the analysed data metadata with the dataset key value pairs. If I had used a different method or different parameters to analyse each image, I would have included the metadata for the analysed data with each image. It is possible to review some of the results in Amero. So here we can see one of the regions of interest that results from the image analysis. The analysis also involved taking some measurements. These measurements are stored in a file attached to each image. Please note for this RDM byte that you don't need to follow this workflow exactly, but this example gives you an idea of how it can work and how you can let Amero do some of the work for you. Please see links to additional resources alongside this video, as well as other RDM bytes in this series.